So this is chapter 14, and chapter 14 has paint, glass, and soil. So, one here. So, I mean, this paint, so this is kind of like a definition of paint. So paint spread onto a surface will dry into a hard film, which that's not very profound. Now, paint, what it consists of are pigments and different additives, and they're suspended in a thing called a binder. One of the most common types of paint that is involved in criminalistics is finishes from cars or automobiles. So automobile manufacturers, usually they have a variety of coatings that they apply to the body of a car or truck. So the coatings include electrocoat primer, the primer surfacer, the base coat, and then the clear coat. So electrocoat primer. Okay, then the primer surfacer, so that's going to make it ready to for the paint to stick, and this has color in it. Okay, it's highly pigmented, and then the base coat. Okay, and this is what the base coat is, what makes your car look pretty. Okay, so that's when you want whatever color you want. Like red or blue or whatever, and then the clear coat goes on top, and the clear coat is going to protect it. Okay, so there's lots and lots of different kinds of automotive paints. So most of the time, the way that they're going to compare them is, guess what? A microscope, right? So a stereoscopic microscope. So they're going to look at color, they're going to look at surface texture, and then they're going to try to look at the layers of colors that are added. Paints it's hard to individualize them unless they have a sufficiently complex layer structure. So they have to be very unique. So it have to be almost like a custom paint job. In the book, it goes into lots of detail about what the components are within the paints, like what about the binders and everything, but I'm happy if you just know the basics. Okay, now, how do we compare paints? So the way that we can do that is two different ways that are really chemists do. So one is called pyrolysis, gas chromatography. So pyro is fire, and lysis is like breaking things apart. And gas chromatography, or chromatography is the right way you're supposed to say it. Gas chromatography, we often refer to it as GC. It is a way to separate molecules so that you can see what's actually in a chemical. Another way is infrared spectrophotometry, which we call IR. So these are things that we do when students learn about it. They usually learn about these in like organic chemistry and beyond. So they're kind of tricky and complex. Okay, now those two things, so GC and IR, are going to be used to look at the binder formulations for the paint. So I have a schematic diagram that explains how pyrolysis GC kind of works. So the idea is that you're going to inject the paint sample into a thing called the pyrolyzer, and that's going to burn the paint up, okay? And at the same time, it injects a gas, okay? So that's where the gas part comes in. So we inject a gas into there, and then the molecules are going to move through a column, and they move through the column according to size. Okay, so usually the smaller molecules are going to move through faster, and as they go through there, they're going to be detected at the end. So we'll be able to detect the chemicals coming off in the gas, and you'll get a little diagram that kind of represents what's actually in there. And the diagram looks like, I don't know, a bunch of mountain peaks, maybe is the best way to describe it. So when you take organic chemistry, you learn how to do this and how to interpret these results. Okay, so solid materials such as paint may be heated or pyrolyzed, okay, so to really high temperatures. So then what's going to happen is they're going to break down or decompose into gas products, and that's going to flow through the column. So it's gas. You're heating them up to their gas. So crime labs are often asked to identify the make and model of a car from a small amount of paint, so they can use color charts for the different finishes and compare them. And then there are some databases that will have like what you're looking for, so you can compare the graphs that you get out of the GC. The pictures are, I remember in organic, they can be kind of tricky to read, but what you're doing is you're trying to compare and see if the graphs are similar. So we have paint pyrograms of acrylic enamel paint. So A is from a Ford and B is from a Chrysler. And what you'll see is different molecules coming off through the detector over time, and they're going to create peaks over time, and the idea is that you can compare the two. So there's definitely differences between the Ford and the Chrysler. Like at around 3 minutes, there's more peaks coming off in the paint that came from the Ford than there is from the Chrysler. So there's different peaks coming off at different times, and that's how you would compare the paints. Okay, so paint collection and preservation. So paint chips are most likely found on or near people or objects that are involved in hit and runs. Okay, so when paint transfer occurs in a hit and run, uncontaminated reference paint should be collected from an undamaged area of the car, and that undamaged area of the car should be close to the damaged area so you can get a good comparison. Usually the paint is collected and it's put in a druggist fold, like it's put on a piece of paper and folded up, or it could be put into glass or plastic vials. So paint that's smeared or embedded on garments, so somebody's clothes or objects, you usually collect the whole object, and it's going to be packaged and sent to the lab so that the lab can get the paint off of the garment, or they can see how the paint is on the garment. Tools that are used to break into buildings or safes often have traces of paint on them. So the tool would be collected, and then you'd also collect reference paint samples. It's like, so whatever you broke into, you collect paint from the tool, and you collect the tool, and you collect some paint from maybe the door frame or the window. Okay, now glass is the next thing. So glass composition. So what is glass? So glass is a hard, brittle compound that's composed of silicon oxides, which that's sand, and it's mixed with different metal oxides. Sodium carbonate or soda baking soda is normally added to the sand, and that's going to lower the point at which it melts, so it'll melt at a lower temperature. Calcium carbonate, which is lime, is also added to the sand, and that prevents the glass from dissolving in water. So forensic analysts, they're going to examine the soda lime used to manufacture window and bottle glass. So they're going to look at the, not necessarily the silicon oxide, the sand, but they're going to look at the soda and the lime that are added to it to determine where the glass came from. Shoot. Oh, I did. Okay. So, molten glass, when molten glass is cooled on a bath, it's cooled on molten tin, and that's called float glass. Okay, then tempered glass is stronger than normal glass, and that's because it's heated and cooled really fast. And then laminated glass is what's in car windshields, and it's a layer of plastic between two pieces of ordinary window glass. So there's just different kinds of glass. float glass, tempered glass, and laminated glass. So glass comparison depends on the need to find and measure the properties that will associate one glass fragment with another. And we want to eliminate other sources, like where else could it come from? So to compare glass fragments, the forensic scientist is going to look at two important physical properties, so density and refractive index. So we'll talk about those here. Okay, so a flotation method. So it's actually a precise and easy way to compare glass densities. So you guys remember doing the density of metals in the very beginning. So in the flotation method, a glass particle is immersed in a liquid, and the density of the liquid is carefully adjusted by the addition of small amounts of an appropriate liquid into the glass chip remains suspended in the liquid. Okay, so we want to add it to there. So at this point, the glass will have the same density as the liquid medium and can be compared to other relevant pieces of glass that if they're more dense, they're going to sink, or they might float, or they might remain suspended. So we'll try to do this flotation method. Okay, now immersion method is a way to determine a glass fragment's refractive index. So the bending of a light wave causes a change in the speed. Okay, so as light is going through a liquid, it bends, okay, and that's due to the change in its speed, and that's called refraction. Okay, so the ratio of the velocity of light in a vacuum to its speed in any medium determines the refractive index of that medium. Okay, so the ratio of the velocity of light in a vacuum to its speed in any medium determines the refractive index of that medium. Okay, so what you do is you take the glass particle and you put it into a liquid medium that has a different refractive index, and you want to find a liquid that has the same refractive index as the glass particle. So I'll show you an example. Okay, now you can do this with a microscope. And so you can take a fragment of glass and you put it on a slide, and then you add different liquids with different refractive indexes. So there's a line, it's called the Beck line. So it's a bright halo that's around the border of a particle that's in a liquid that has a different refractive index. I'll try to show you how this works in lab. Okay, so at the point where it matches, the Beck line is going to disappear, and you can't really see any contrast between liquid and the glass. Okay, now analyzing cracks in glass. The penetration of window glass by a projectile, it could be a bullet, it could be a stone. Okay, whenever something goes through the window glass, it's going to make cracks that radiate outwards, and they call those radial fractures, and they're going to go around the hole, and that's called a concentric fracture. So radial, like kind of radiate like the sun, like the rays of the sun, and encircling the hole, they're going to go around in circles, maybe like a bullseye. Okay, so they can look at the radial and concentric fracture patterns in glass, and they can determine the direction of the impact. So how did the bullet or how did the rock hit the window by looking at the patterns. So this shows glass and something hitting it. Okay, so like a bullet, say a bullet hitting it. So at first, what's going to happen is the radial cracks are formed. Okay, so the cracks are going to form on the side opposite where it's being hit. Okay, so it's going to get hit on one side and the cracks will form on the opposite side. Concentric cracks occur outward. Okay, so you'll get circles and they're going to go out from the hole, and they start at the same side as the force. Okay, so the radial cracks are going to start on the opposite side, but concentric cracks will start on the same side as of the force. Okay, so a bullet often is going to leave a hole that's wider at the exit site. Okay, so it's going to be bigger where it's exiting. And you can use that to determine the direction of the impact, where the bullet came from. Okay, so the direction of impact can also be accomplished by applying this rule. It's called the 3R. So radial cracks form a right angle on the reverse side of the force. So that's the 3R's. So radial cracks form a right angle on the reverse side of the force. So the sequence of impacts when there have been successive penetrations of the glass. So if the glass is being hit over and over and over again, like maybe it's an automatic weapon and they're shooting it a bunch of times. Okay, so if that's happening, it's sometimes they can determine, so the sequence of impacts when there have been successive penetrations, it's possible to determine the sequence or the order that the impacts occur, because a fracture always terminates at an existing line of a fracture. So it would be like a puzzle. So a fracture can't continue if it comes into another fracture. Okay, so 2 bullet holes. So the left hole preceded the right hole. So if you look at this, so there's a bullet hole in the center, okay, and then there's radial lines coming out. So like cracks radiating from the center of the hole. And then there's a concentric circle. So in order to figure out that this one occurred first, okay, there are radiating lines, and they're extending out further than the ones on the right side. The right side ones end where the left side ones already are. Okay, now to collect glass. Okay, so if there is a possibility that the glass fragments could be put together, then you have to try to collect all the glass. When an individual fit is thought improbable, the evidence collector must submit all of the glass evidence found in the possession of the suspect, along with the sample of the broken glass that might be at the scene. Okay. Okay, so the glass fragments should be packed in solid containers so that they don't break anymore. If the suspect's shoes or clothing are to be examined for the presence of glass, they should be wrapped in paper individually and taken to the lab that way. Okay, and then the next thing is soil. So soil's everywhere, right? And it can spread. So, you get soil, dirt on your shoes, right? And you tramp around and you go someplace, you're going to spread it with you. So soil can be transferred from the scene, between the scene and the criminal. So most soils can be differentiated or distinguished by what they look like, so their gross appearance. So we have a lot of clay around here compared to other places. So typically a side-by-side comparison of the color and texture of the soil specimens is easy to perform, and it can help you figure out where the soil came from. Okay, so there are people called forensic geologists, and they're going to look at the soil and try to figure out what kind of minerals are in them. So considering the vast variety of minerals and rocks and the possible presence of like artificial debris in the soil, the forensics geologist is often, they have lots of things to compare. Standard and reference soils are to be collected at various intervals within a 100-foot radius of the crime scene, as well as at the site of the crime, okay, because they want to compare it to the question soil. Soil found on the suspect, if it's on the shoes or the garment, shouldn't be like picked off of the shoes or the garment. It should be wrapped up and you should take it to the lab that way.